And you can start whenever you're ready.
Okay, I think we might have a couple more.
Okay.
Does anybody have the time?
You're over. Go ahead.
Oh, okay. 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 Hi, everybody. I'm Amanda Borens. I'm the Platform Architect and Development Team Lead at Critical Path Institute. So I think a lot of you know Rick Liskey. He's been promoted to be our Chief Technical Officer. So I'm taking over his role. And today I'm going to give you all an overview of the ReSeq-TB platform. Can I just ask everybody to raise hands if you already have an account for ReSeq-TB? And how many of you have contributed data to the platform? And then lastly, how many people have never seen it before? Okay, so I just wanted to get an idea so that I didn't linger on topics too long. It seems like we have a pretty good blend of people. The first thing that I want to show you is the web address. If you Google ReSeq-TB, relational sequencing tuberculosis, basically, if you Google it, you're going to find this website. And if you'd like to make a note, it's there at the top. It's platform.reseqtb.org. What you see at the landing page, I've scrolled down just a bit. And what I'm showing you, though, is that we always have a count of all of the tuberculosis isolates that we have in our database. And this count is reflective of isolates that have both genotypic and phenotypic data. So on many occasions, we get sequences, but they don't have matching microbiological testing data. And sometimes we get, actually, believe it or not, we get testing data, but the sequence doesn't make it into the platform because it fails to pass QC. So what you see on this page is always a count where you have both pieces of information available to you. As you can see, we have had quite a jump in the last month or so since I've joined. And I've really been pushing our team to go back and make sure that they've got all the backlog of isolates uploaded. We've been working on an effort in parallel that has slowed things down somewhat, which is to include all variances, all SNPs, at all loci across the whole genome. And so that is happening in parallel. But for now, what you're going to see is the platform with a filtered list of genes of interest for drugs that have been tested. Okay? So from that data, what you'll see is that we have about 4,500 isolates, and about a third of those, about a little more than a fourth of those are resistant in some way to some drug. And I'll just clarify that the way that I've counted these as MDR, XDR, pre-XDR fluoroquinolones and injectables, all of those are counted in such a way that if any result is resistant, it falls into that category. And I'm clarifying because it may not be obvious that on many occasions we get more than one result for the same drug. So there are labs where they have done DST as well as a line probe assay, and sometimes even an MIC as well. So in some cases we have 3 results for the same drug. So it's a richness of data, but it makes things complicated, obviously, when you're counting and chopping it up. The other thing I want to point out is that we have really lately increased quite a bit in the representation geographically and among lineages. So in the past, we had a shorter list of lineages represented in our database, and we had a smaller number of countries represented. So there are probably more countries represented than you see here, because, and those dot sizes are scaled based on the number of isolates from that nation, right? We are careful because we are an organization that also manages patient clinical data. So we're very careful about not being able to trace the data back to the location of the patient. So the best that we will do is to indicate the country that that isolate came from. But on occasions where the data comes in and we know that it all came from London, for example, that probably wasn't always where the patient actually had the case of TB. So in that case, we leave that blank. If we can't trace it back to the actual origin of the patient in question, then we don't put a country. So you're probably seeing less geographic diversity than actually exists in this database. Okay, so with all that said,
So you just leave it blank, or do you put where it's from with an S?
no. So there's a table, a column in the database that is for country. If we know the country, we enter it there. If we can't be sure of the country, we leave it blank. And when I say we can't be sure, I mean we probably don't know at all. So there are times when we have studies where the contributor knows the quality of the isolate and the collection methods, but they may have had labs processing it at multiple locations, and it just gets more difficult to tie the actual location of the individual isolate back to a particular country. So now I have turned off, for the purpose of this demonstration, our two-factor authentication. But if you already have an account on ReSeq-TB, you know that we ask you to set up a token system on your smartphone or computer. And that will give you, when you try to log in, it will say, give us a token. It will email you, or it will send it to your smartphone. You enter the token, and that's the second factor of making sure that you are who you say you are. So I'll just log in. I have probably more access than you will see because I am a user that may be uploading data that I'm contributing. So I have two choices here. I have the upload repository, but I also have the actual database itself. And today we're talking about the actual database. So if you don't see these choices when you log into your account, that doesn't mean anything except that you're not actively uploading isolate data. Our connection here seems to be considerably slower than I'm accustomed to. So I would warn you actually that your internet connection will have an effect on how quickly all of these data load. So the first thing I wanted to show you, especially for the folks who haven't used it before, is that you always have the user guide, the full user guide available here on the platform. If you click on this resources at the top, notice that you have three major choices, categories of options to connect to. One of them is resources. It lists both a user guide as well as general information. And the user guide is where you're going to find things like explaining two-factor authentication, your terms and conditions for using the database, things like that. And then even more specific information about actually browsing the data and about using the visual browser. But I'm going to give you a tour of all of those things, so I won't go into a lot of the details. One of the things that I want to point out, though, really quickly, is that under general information, you have expanded links to things that might be useful in understanding the context of the data, so not necessarily related specifically to ReSeq-TB, but for example, information about the CDISC DTM standard, which is a big part of what we do at Critical Path. And some of the names of the database columns in our database are reflective of some of that. So that might be helpful. And then the full list of domains, which is basically a list of the column names and what they are in each of the tables. And then FAQs, just about, that's probably a good place to go if you've forgotten how to quickly do something. And then the last thing before I get into the meat of the data is to show you this little gear, which is our settings. And under settings, you can change your password. You can apply for a different database. So it's probably good to point out that Critical Path manages consortia across many diseases. And so you could apply for a different database here. Two-factor authentication, giving you the option to set it up and instructions for installing the app. Change your email address, and then here's one I really want to call your attention to, and that's the help and feedback. So I think it's worth mentioning again, the gear, help and feedback. So I'm using Rick's account, but that's obviously not the email address that it would go from if it were you, right? It would be your email address that you've signed up to register for an account to access ReSeq-TB. And I really encourage people to use this web form. Every single day, we have staff that check this email account, and we definitely will take your feedback. So I think one of the things that I have noticed, because I have a life sciences background and I've worked with genomics data before, I've noticed that there are tools that I would like to use that don't yet exist. And so I think that what this platform needs is feedback from all of you about what you would like to see. Not just, I hope that you won't access it and say, oh, it doesn't do this thing, so I won't use it. I'd really like to hear exactly what it is that you'd like to see in ReSeq-TB. The last little housekeeping thing here is, if you go to apply for a database, in order to do that, before you can send off your application to this person who checks the email account, you'll have to accept these terms and conditions. And I think that if you are considering accessing the data, but also if you're considering contributing the data, it's good to know some of the terms and conditions that we've defined. And some of them are things like, if you plan to publish based on any of these data, you will give us the right to review the publication to make sure that the analysis fits with, the data that we have. And also, because there are times when data changes, because, for example, we have modified the pipeline and we have different annotation. We would want to make sure that your data reflects the latest version that we have in the database.
Can I ask a question?
Yes, sure.
I'm sorry if I missed it. One of the things that you just said in the last few minutes, you have to have an account to get that information, or that's in the open area?
Just you have to have an account to log into ReSeq-TB, which I've already done.
But why would you not have some of the information not on your password? Why like the user guide and
Oh,
the
interesting.
application and the
Yeah.
line? I mean, like if you just go there to the first page, you know nothing about this.
No, I think that's a good point. Yes.
Before you actually, now if you want to access the database, I can see that you will have to have an account.
Yes.
But if you want to promote this, you would like it to be.
Thank you so much. No, it's a fair point. It's a fair point. I think when it was set up this way, it was because it was assuming that you all are already accessing these data, and maybe you've forgotten how to do X thing. So, but I think it's a fair point, and I'll definitely take your feedback, and I will use it to the best of my ability. We have a lot of work going on right now. That are about making this more user-friendly, and that's exactly the category of things that I'm looking for. Okay, so I will jump right in now. So assuming that you have an account already, you're in the system, the first thing that I want to show you is the data access data browser. Okay. So I think it's a good time for me to give you a general overview of the data that you're going to find in ReSeq-TB. I think I gave you, so you'll see two items in this drop-down list. I gave you an overview that we have genotypic and phenotypic data, but I'll be more specific now. What I'm about to review for you are the two categories of data. One we call microbiology and the other pharmacogenomics. And those come from some of our SDTM and CDISC standards. So they're long names, and we export them as PFF and MSF files. So you can always remember microbiology as M. So the category of microbiology, like I mentioned briefly, has test results for each of these isolates. They may have tested 2 or 3 drugs, or they may have tested 10 drugs. So it's variable from isolate to isolate. But what you will see has been curated and checked and definitely made sure to be accurate and can be traced back to the contributor's lab. And then you will only see microbiological data if that isolate also has genomic data. So the genomic data that you'll see is not the actual raw sequences. We take in FASTQ files. Matthew there runs them through an extensive pipeline. We produce a SNP report. That SNP report gets mapped into our database, and the isolate ID itself matches up with a record or multiple records of microbiology testing results, okay? So with that context, I'll show you the 2 files, starting with pharmacogenomics, just because that's the default here. There are many columns, and as a researcher, if that is your area of interest, you will not need all of these pieces of information, right? Many of them are of interest if you've drilled down into a particular set of records that you're looking at. Some of them are for provenance. Some of them are for identifying it, and tying it together with other things. So the first thing that I want to show you when using the data browser is something that we call roll-up. So the projection is not exactly clear, is it? So there's a hyperlink there at the top of the table indicating roll-up. And what you see there that you sadly cannot read is...
read any of it, actually.
You get the general idea, I'm sorry. So each of these are column names within the table. So if anybody has experience with Access or any sort of basic database, every record is a row in a spreadsheet, right? And every column is a field in a database. So these are the field or column names. And for pharmacogenomics, I want to show you an amino acid change. You'll have to take my word for it. A gene name, and a nucleotide change. And when I do that, and I scroll to the bottom of the column list, I can activate rollup or clear rollup. If I've changed my mind and I just want to back out, I can clear it. Or I click activate rollup, and you see this becomes a much more concise view. But what I really like about this is that it also counts for you. So for every gene and mutation, you have a count of the instances in the database. And that's true for all of the other fields. The next thing I'll show you, even with this grouped and counted list, I can add filters. So this first column here is the nucleotide change. The second column is the amino acid change. And then the last one is the gene name. So let's say I'm really interested in looking at RPOB. My list is filtered by that particular gene. And so I'm seeing counts of all of the mutations in that particular gene. And I also know that I have 254 records of mutations in that particular gene. The other thing that I think is important is that there are these little arrows. You can kind of get the idea that there are arrows. And when I click on those, it will sort ascending or descending, so A to Z or Z to A. And basically, I want to show you the same thing with phenotypic data. So again, you'll see more columns than we really need. And they are things like, now that I know that you can't read any of it, there are things like the study that it came from, so there's an identifier for the study. The domain, meaning that this is an SDTM CDIS thing. The domain is that it's microbiology susceptibility, and then some IDs that tie this isolate back to the phenotypic, or the genomic data rather. And then other things like the fact that it was an isolate and test results. And I'm going to consolidate that in the same way by using the roll-up data feature. And under the roll-up data feature, you'll see I'm selecting MS drug, MS res, that's the result, and MS test code. And remember, back to the fact that under the resources that I showed you in the beginning, there's a domain list if you ever needed to have help figuring out which column it was that you're looking for. So then when I activate rollup, I get for every drug, and I'll sort them. So for every drug, can you kind of tell that we have amicase in there? And then we have both DST and MIC results. So the way that I've selected these particular columns is causing it to count for every drug and test method how many resistant and how many susceptible, and if applicable, how many intermediate or borderline results I had. And so that's a pretty useful and quick.
So, ma'am?
Yes.
So as part of your curation, if they don't match, you don't include them? In other words, the MICs, you have 2 different methods, right? If they're discordant, is it put in there?
Oh, no, we keep them.
Right, but you show them, or?
So they will be available. You'll see them here. You can filter it however you like if you don't want to use line probe assay. So for example, I've been working on an analysis recently, and one of the things that we did is we decided we prefer a liquid culture, we prefer DST if we have it, but if MIC is the only thing we have, we'll take it. We do not want to use line probe assay. So we keep all of it because you may have different goals. You may even want to ask the question, how often is this discordant with that? And how often is that associated with a particular mutation, right? So it's information, and we don't want to filter anything out. If we have it and we can verify that it's valid, then we keep it. What I want to end with here is that you always see found 71 results in 25 milliseconds. Gives you kind of a nice listing of the stats. But what I want to end with here is that there isn't a functionality yet, and I see this as a big gap. So it's one of my first projects, is that I want you to be able to export that data after you've filtered it and sorted it and grouped it, however it is that you like. And presently, this is just for viewing. Okay, so that's something that will be coming for sure. Now that also leads me into a couple of other things that I want to add functionality to, and that's when I show filters, I want to be able to only look at DST, right? So that's what we were talking about. So if I filter by DST, I get this list of DST. What I don't have, though, is a list of the possible entries in that particular field. So I want to add a drop-down list so that instead of typing DST, because maybe you don't know exactly how we've codified it, I wanted to show you that there are DST and MIC and LPA values in that field. That way you can filter it a little bit more intuitively. And then the last thing I wanted to mention is that presently these do not roll up the data together. And the reason that that wasn't tackled in our first run of this is that we have, I think, something like 80,000 records of SNPs. That's based solely on the filtered list, so it will be much, much, much longer for the same number of isolates when we finished our all mutations database. And then we also have about 30,000 records of phenotypic testing results. So the way that these work when you join them together is that you have to multiply 70,000 by 30,000. And so you're talking about hundreds of millions of possible combinations. So I think what I would happily take feedback about this particular thing, but I think what would be useful is if you could export those in a joined fashion. So if you said, I want to filter it, and I only want to see amikacin results, and I only want to see them for DST, then that will reduce the number of records so much that you would then be able to join all of the mutations with each of those results. But think about that, and if you have any feedback about what might be useful as a researcher, I would happily take it. And so that is the data browser, which again we found under data access. What you can do presently is that if you go under data access and data export, you'll see that we have those same splash page stats. Those are available to anyone without logging in on the splash page. But what you have here in this version is you have exports. So if you use SAS for statistical analysis, you can export it as an XBT file, or you can export it as a comma-separated file, which is comma-separated value CSV. And what those do is they export all of the data that are publicly available for both of those tables that I've just shown you. So if you are interested and you have the ability to combine those things, then you can easily export those files here and then join them up however you like. Meaning filtering them first and doing whatever sort of limitations that you want on the list of records. The other thing that I would show you from the export that you can get now if you have an account is this drug resistance and associated mutations report. So something that has been an ongoing effort is statistical analyses that predict which of these mutations are are associated with resistance to certain drugs. So this version is filtered by a list from our expert panel of genes and drug combinations of interest, right? So this isn't every possible mutation in the genome associated with every possible drug, but it's still a really extensive list of results. And so if you export that file, you'll get all of the likelihood ratios and the p-values and the confidence intervals for each of these mutations, which is a good segue into the last category of data, which is the visual browser. How many folks in this room have used the visual browser? Good, actually I'm surprised. So this is something that is currently being developed, so it's being enhanced as I speak. There are open tickets with developers working on this. But what you'll see is a list to the left of the drug results and a list to the right of the mutations. And presently, in the screen, this is specifically the genes or the loci of interest, okay? And when I uncheck this, so let's just start with something obvious, like rifampicin. So, I know now that there's one gene of interest for rifampicin, and that's RPOB. There's a little plus sign that you may not be able to see, but when I click on this plus sign, it gives me a complete list of all of the mutations that we've seen in ReSeq-TB with that particular gene. And you'll notice a green dot under the LR plus value. If I'm lucky, there will be other colors. Yeah, look at that. Okay, so the green and the red and the yellow indicate something with the statistical significance. So if you look here under the LR plus filter, you can check and uncheck which category of significance you're looking for. And then, actually, this is a good time for me to point out as well. There's a little question mark here that tells me high confidence is an LR plus greater than or equal to 10. Moderate confidence, or yellow, is 5, less than or equal to less than 10, but greater than or equal to 5. And then low confidence is less than 5, but greater than or equal to 1. And then there's a none category. Yes?
Yes, so this level of confidence is just about the frequency of that particular mutation in the ReSeq-TB database.
So the ticket that I have open right now is that these green, red, and yellow indicators will be hollow if the p-value is not significant, which is very often, you don't know. It might have a particular LR plus value, but you only have a couple of resistant isolates. So that you wouldn't want to rule it out necessarily, you just don't have enough information yet. So the hollow circle will indicate that the p-value is greater than 0.05, just a random cutoff for the sake of display. But if the p-value is significant, so less than or equal to 0.05, it will be a solid green, yellow, or red dot. One of the other things that you see.
So yes. What happens if you have a proton that has high confidence and you get a new data set that says that mutation now is susceptible? And let's say you only had one example. So now you have two examples. One says that mutation is successful, and one says you just go to literature or to what do you use as your gold standard?
Well, I think if you have worked with statistics, you know that having two examples may or may not actually tell you anything. So from my perspective, if I'm looking at it, I'm not saying it's not associated, but I just think the case is still out. The jury is still out on that particular one. I might not remove it from my list of interesting mutations, but I probably wouldn't publish anything based on saying that is significant because I only have one and it was resistant, right? So this just lets you decide what your criteria are. Arbitrarily sometimes with minimal numbers of isolates in a particular drug category, we have to just say we'll take fewer isolates and say that might be interesting, right? But some of them are tested every single time. I mean, I can tell you that that almost every isolate has a rifampicin result and an isoniazid result, right? Ciprofloxacin very rarely relatively speaking, has a result. So that's one where, if you want to look at ciprofloxacin, you kind of have to work with what you have, but you have to use your judgment as a scientist to decide whether or not that's significant. So the first thing that I showed you was the list of drugs, which narrows it down to a list of genes of interest. And then I have an expanded list of mutations. I can filter that list here by checking and unchecking. Let's say I only want to see those with an LR plus value that are significant, so greater than 10, greater than or equal to 10, rather. And what I will see is that the mutations list does not change here, but that there is a filtered list at the bottom of my screen that does change. So all of the filtering happens down at the bottom of my page. So I'm just taking a moment to note that. But for the sake of this demo, I'm going to show you that I can uncheck everything and look specifically at a couple of interesting ones. Okay? And then once I click these little zoom, see what happens in the radial graph? I hope that's clear. I can also hide this, by the way, to make more room for the radial graph. So this is the radial graph. Each of the orange bars indicate a particular gene of interest. And everything went gray because I focused solely on this particular set of locations that are very close together. So I can change my focus here. So what is black is going to be in my filtered list, and what is gray is excluded from consideration right now. And then if I look here, I have the ability to zoom in and zoom out. So I'm going to just okay. So in this linear graph, I've zoomed in to the particular mutations that I'm interested in looking at, and what I see is green and blue. Okay, and the orange above is different loci of interest, but the particular gene that I want to look at, you can see that when I hover over the green or the blue, I get information there in a little pop-up window. If I click on it, that information becomes static and on the right side of the screen. So right now, I'm looking at drug rifampicin, gene, RPOB. I have the start and stop position, or start and end position, and the LR plus value along with the actual nucleotide change and the amino acid change at that location. And then what I have here, which I think is really useful, is the number of true positives, false positives, true negatives, and false negatives. And obviously we use that to calculate the sensitivity and the specificity, which then becomes the likelihood ratio. And all of the mutations in the database, all of the occurrences of that mutation in the database are listed here in my filtered table. Now what I like about this new feature is that unlike the roll-ups that I worked on in the data browser, I can actually export this to a CSV. So once I've done some browsing on the genome and I'm looking at a particular gene and a location of interest in a particular set of mutations that I've sort of filtered out based on their LR plus values or whatever criteria you'd like to use, then I can export the result of that filtered list. Yes?
Yeah, so to ask to determine the sensitivity or specificity, are you comparing against phenotypic DST? And it's a fault. Okay, and so then if you're using phenotypic DST as a database, capture which method you use and which critical concentration.
Yes.
Yeah, okay, so that's all.
It's all in the phenotypic data. So what you're seeing here is the result of the analysis, but it's the combination of the genotypic and the phenotypic data. The phenotypic data has the test method, the results, the critical concentration or concentration depending on what's appropriate. It also says whether it's a liquid culture or solid culture or molecular method, right? So all of these data are definitely available. So the other thing that you'll see here is that this is a different format. This list of records that you see on this row here, it is not an export of either the genotypic or phenotypic. It's a consolidation and summarization of a particular mutation associated with a particular drug. So the expanded data that you just asked me about was considered in the analysis that produced this list, but it is also available if you export the full phenotypic data files. So I think something that I would have found interesting if I were doing this using ReSeq-TB is that if I had a particular mutation of interest, I could export the phenotypic and genotypic data. I could filter the list of genotypic data by just that mutation, even with Excel. And then I could go and look for those isolate IDs in the phenotypic list. And then I can look for myself to make sure that I like the way that the analysis was done. If ever you found anything that didn't look reasonable, please do let me know. We have a pretty good process that has been refined over the last few months, but always with statistics, people are making decisions, so feedback is important as well. I think the next thing that I'll do is just, for variety, I'll show you. Again, there's a little question mark in the top right corner of each of these boxes. So for the radial graph, the linear graph, and also this selection of items, there's always help available. And then I'm going to change my selection of drugs. So I want to include amikacin and not amikacin this time. And then RRS becomes the location of interest, and I have a list of 3 mutations of interest in that particular gene. And you can see that 2 have a pretty high LR plus value, and 1 has probably a low one. And then the thing that I have on my list here, is that I'd like to see the p-value right in the table. And I don't believe that we're showing it here. Does everybody see my mouse moving there? This does not include the p-value. It includes the sensitivity and specificity, but not the associated p-value or confidence interval. And we have it. It's in this table, so I'd like to display it. Does anybody think there's any reason not to display it? Right. I think that would go into my decision making for filtering. So at this particular location, I have this filtered list of genes, and then if I export them to CSV, you see that it automatically just dumps it into my download directory. These are really easy to work with in my experience. But then, again, let's review that I can minimize this little window. I can open it and close it as I wish, and then I can highlight that particular mutation and get more information in those boxes to the right. What I haven't shown you is that these bars, the height of these bars, indicates a count of mutations at that location. And the green indicates the number of resistant isolates with a mutation there, and the blue indicates the number of susceptible isolates with a mutation there. And here's where it gets even more complicated. There are shades of blue and shades of green, because sometimes you have multiple mutations at the same location. So we've classified them in that way. If that were the case, if I were you, if I were reviewing these data and I wanted to explore a particular mutation, I would filter that list to that particular gene, and then I would just export the CSV and play with it myself. It's just a little bit hard visually to display that much information in a small spot. And so I think it's worth reminding everyone also that I can always go back to data export and analysis and log back in because I was too talky. Let's see. Okay, so data export and analysis allows me to export both the genotypic and phenotypic files, everything. I can filter it however I like. And then the data analysis result, that allows me to export all of the statistical analysis results of the genes of interest and drugs, drug-gene combinations, right? And I think this particular file, for the sake of showing it, I think it has, let's see how many. 87 records. And as I mentioned, they all have sensitivity, specificity, odds ratio, p-value, LR plus. You know, they have all of the results of the statistical analysis. We don't show everything in the visual browser, but it's all available for export here. And you can decide what the criteria are that we want to use. So, what's that?
No, certainly, you can see it.
Oh. Well, how about this? Just everybody that has a question, I'm very happy to take whatever question. We can go back and look at anything we need to see.
So, in the factor-resistant true-out gene, there are examples of a strain having 2 mutations in the same strain, MDR-TB. How do you actually, is it on the same line, two, or is it putting us up? How do you actually?
There'll be a separate line. So multiple mutations in the same gene, each mutation will have its own line. And in fact, I think if I'm not mistaken, there are times when there's a nucleotide, the same nucleotide change might result in a different amino acid change. So you might have, you'll have a record for the significance of each occurrence. Yes.
And you can differentiate mutations in hotspot regions versus in the rest of the
Yeah.
gene.
So you'll have the location of the nucleotide change. You'll have a position there in the data. I'll show you once it's really clear. And you can filter it appropriately. Yes.
There are a number of other databases out there, and is this meant to kind of be the mother of all databases in terms of drug resistance genes? And I mean, I'm just trying to put this into context. There's this over there, and there's that over there, there's a paper here, and a meta-analysis there, and it's really quite confusing.
Joining database. Okay,
I
so
think the one thing is.
So I'm going to plug the breakout.
There's no EIS mutations on this.
not in this particular list, right?
Yeah, and there needs to be.
So the analysis that I've been working on from these data in ReSeq-TB is a full list of other things that weren't already defined by the expert panel. And so you have that ability by exporting these data. That's the first thing I'll say. The second thing I'll say is that you can always give feedback to the expert panel to say, I think this is a particular mutation of interest for this particular drug. I think that would be considered. And then in relation to that, this is an ongoing and ever-changing list of genes of interest for particular drugs. Yes? It's not, it is better, actually. Thank you.
Much better. Thank you.
So to finish that question,
But should this just be a database, not like where the data is what the data is? It's matched to the standard data with the phenotypic data, and you should have a sensitivity, a likelihood ratio, an odds ratio, and it shouldn't matter what the expert panel thinks. It should be a mutation on the list.
yes.
Why?
So possibly because of what I mentioned, that there are hundreds of millions of combinations of drugs and mutations. So just from a standpoint of running all of these statistical analyses on every possible combination of drugs and mutations. I am separately working on something that is a more extensive list so I have a list of genes for each drug, and I'm looking at every mutation in that gene for significance. And that's something that anybody can do by accessing these data. And then the list of genes of interest is ever growing. And then I can tell you from firsthand experience that we're hoping that these data, as the number of isolates grow, will be informing the list, as opposed to having an expert panel say, here's a list of drugs and genes that we think are significant. We would like to get to the point where we can run this analysis every so often, because it's such a big and monstrous thing to do for that many possible combinations. But we would like to get to the point where we're able to generate and tell you, we suggest that these might be significant mutations that might be conferring resistance to a particular drug. So that's something that we're heading toward. It's a really big project. Even just running the analysis is a pretty time-consuming thing when we're talking about every possible combination. And I think you had a question.
Yeah, about new drugs. For example, there have been
about bedaquiline.
Yeah, just as an example.
So,
So the ATP.
yeah, I don't remember, I won't say with confidence, I don't remember seeing a bedaquiline result yet in the database. We have almost 1,000 isolates being currently processed right now in Critical Path Institute by the data management team, so that could be changing imminently. But if anyone has bedaquiline data, this is a great time to offer it.
The issue is that for these drugs, the new drugs, you have cutoffs clinically established, so you will have MIC data
Right.
more often than
Yes.
So,
But it's information,
yeah.
right? I'm sorry I didn't actually finish the answer to your question, which is that in regard to whether this is the database. I think we would like it to be the database. There's been a tremendous amount of effort involved in building a repository that would work for everyone. We're very open to feedback and changes. But then I also will plug for the breakout session tomorrow afternoon around this same time. We have a breakout session about database interoperability. And so what we're working toward is, right now we have about 5,000 isolates. We're adding, as like I said, imminently about 1,000 isolates. We're always working on trying to get more isolates into our database. We also have a pipeline running where we have a funded project to sequence isolates for labs that have phenotypic test results but did not have the funding or resources to sequence those particular isolates, and they're pulling them out of the freezer for us and they're sending them off for sequencing, yes.
So maybe another way of asking the same question is, how different is this particular database from other existing, pretty extensive databases?
Yes, no, I think the thing that makes this one different is that as a scientist, I'm confident that these things have been checked and are definitely curated. What I'm not always confident about is some of the places where I can go to maybe get a list of sequences from NCBI or GenBank. Anyone can upload sequences, right? And the other thing that really makes a big difference is that every single one of these has phenotypic results. So that's where the real power is in combining the fact that it was resistant with the fact that it has these mutations, as well as all the other records where there wasn't a mutation, but it was also resistant. So you have the analysis
You
coverage.
also have metadata too.
Yes, for all
And
of
that's
them.
the biggest difference, I think, the difference between, they're trying to integrate the clinical information with the strain, whereas the TB Dream database and others,
Yeah.
are focusing on the strains and resistance and genotype. So I think it's the clinical information along with
The power of
genotype
that
and
together.
phenotype.
Yeah. And then because I'm working on this interoperability question, I think there are others that would like to submit to us as opposed to taking, taking it in the other direction. But I could be wrong about that. I think there would be nothing wrong with having multiple databases with all of everyone's isolates available, right? Multiple sources. So we're not opposed to that idea in principle. But the thing that I have found is that people often have genotypic data or phenotypic data. They don't have both of them. And then the other point about curation, the last thing that I'll say about it is that we also do a pretty extensive checking process because people have identifiers for their isolates, but they've also shared that set of isolates with another lab in another country, and they're calling them something different. So they look different, but we do pretty extensive process of investigating the data with the contributor, having conversations on the phone before we ever even see the data. And we make sure that we're not repeating the same isolate from two different sources. So there's an extensive amount of data curation that goes on even before we look at the data. If there's ever a question about a particular record, if, for example, we don't have the critical concentration and we can't actually be sure of this result, we go back and we visit with those people. Yes?
So just to answer the question that came up, the distinction between the genes of interest list and the CSV that's there, while you've been talking, I just downloaded your CSVs, and the ATPE gene, for example, you have to look for it as RB1305, but you have 29 isolates in the list that is currently downloadable that have mutations in RB1305. So it's there, it's just not on the genes of interest list.
Yes. So that was my point about all of the hundreds of millions of possible combinations. And I think that's why we've been really open with, we are giving you some suggestions of analytics, we're showing you some of the statistical power of the data, but But we definitely encourage you to take all of it and do whatever it is that you need to do to answer your questions. Yes?
So the plans to make the inline sequence there, or the inline sequence there available?
The FASTQ files, is that what you're referring to? Okay, so the FASTQ files and all of the associated products from Matthew's pipeline are all stored on the cloud with the database. So if we have, if you have an account and you have the ability to download data, I will show you that actually presently. What I would like to point out though is that you will see some differences in the counts. So this file that I'm showing you here is an analysis that I showed you with the statistical analysis. The summary of the data is acceptable to our contributors as far as even if they are not yet ready to publish the sequences, they will allow us to summarize the data. So don't be concerned if you download a full export of the database and you see a different number of isolates than you're seeing in this statistical analysis. So if data has been embargoed, so for example, we have one that will be openly available starting in August, that's a pretty typical option that we give to contributors. If they're busy doing work to run analyses and they want to publish, we give them that respect to allow them to finish their work before we make it publicly available. If it's publicly available, you can download not only the data in this curated format that's all been mapped and nicely, formatted into exactly the same columns every time, but you can also download the FASTQ files. And so under data repository, two categories: file listing, which is extensive and probably not what I'm going to start with, or projects. So let's say, so for security, you get logged out obviously if you've been standing in front of the room talking. So under projects, you have a listing of projects and folders. It tells you who created it. These are always going to be data managers associated with Critical Path Institute. But the projects themselves have these little view files icons. Each file, each project. Then when I click on view files, well, that's a bad example. So if that particular project had been uploaded with publicly available data, all of the FASTQ files that are in a zipped format, so the .gz extensions, but all of the FASTQs are available if they're publicly, if they've not been embargoed, they're available. And if you ever had any concerns or questions because you couldn't find the particular FASTQ files that you wanted, then you could absolutely contact me and I can help you find what you need. It's been a really big effort. I think everybody here probably has some idea of how big a FASTQ file is, and so we have thousands of FASTQ files. It's a big effort to store them and also make them accessible and then have them appropriately referenced by each project and things like that. So it may not always be obvious, and I'm happy to help find the FASTQ files that you need. Any other questions? Yes?
Yes, does the database also include genotyping information, remove the MDR profiles for each of the strains, so that you can look at the diversity of lineages of strains in the database?
So I'll show you what we do have for the genotypic data, because that's a good way to go back. Now that you've had a general overview, it might be a little bit more, and maybe you can read it a little bit better now that we've had some IT help. So the genotypic data will have lineage, which is here. And actually, you know what, it'll be easier if I roll it up. So roll up data. Lineage. And then S lineage, which is sublineage. So these, if I sort them properly, and then I think I can go and make the page size bigger. Does everybody see what I'm doing? Just so they fit on one page, I don't have to page through. These are all of the lineages and sublineages that are represented by all of the isolates in our database, but each of those pieces of information is in every record for every isolate. So the possible lineages are 1 through 7, But then we have another one that may not be obvious called 888. It was because of a database technical need to have an integer that indicates bovine, but the sublineage will always say BOV for bovine.
You could probably go to spot type. You could pull out spot types and probably be an MDR. Spot types you definitely can pull out now, I think. It's quite a low bias in that. So I think we can easily find that with another sort of line system.
So, or even a limited mirror again.
Yeah, limited mirror and spot type together would give you a little more resolution, I agree.
So next week we're having a call to talk about homoplacy and trying to use lineage and sublineage as a surrogate for the actual phylogenetic tree that we would. And we want to see if we can find a shorter, lower hanging fruit to be able to identify which of these mutations might be just markers of a particular lineage as opposed to right, yeah. So so it's something that we're mindful of because we're trying to use it.
Oh, very good.
Any other questions? Great. Yes.
One question. Just because I'm not totally familiar with the scope of who can put the genomic, the information in, is this linked up with like CDC or broader?
No, we keep it completely neutral on purpose because we have contributors from all over the globe, and most of them don't want to be associated with American government agencies, right? So we're not associated with any particular government agency. That's the unique ability that Critical Path has. We specifically put it on Amazon Web Services so that it's also not stored in our office. It's stored in a neutral location. I think that's actually helped us to get contributions from people, and it makes some countries more comfortable sharing data with people from other countries that they might not directly share with. Anything else? Thanks, everybody.
We currently have a break, and you have till 3:45, so an extra 10 minutes to be